Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The diagram shows a section through a mitochondrion. R is the area outside the mitochondrion, the cytoplasm, where glycolysis occurs. S is the intermembrane space, which is between the outer and inner mitochondrial membranes. This is where protons are pumped to create an electrochemical gradient during the electron transport chain. U is the mitochondrial matrix, inside the mitochondrion, where the Krebs cycle occurs. T is the cristae, or inner mitochondrial membrane, where the electron transport chain takes place. This membrane is folded to increase its surface area. The electron transport chain generates a large amount of ATP, the cell's main energy source. It accomplishes this by transferring high-energy electrons from molecules like NADH and FADH. As these electrons move through the chain, a proton gradient is established. Ultimately, the electrons are passed to oxygen, which acts as the final electron acceptor, combining with hydrogen ions to produce water. High-energy molecules, such as ATP, GTP, and NADH, are compounds that store and transfer energy within cells, often through the breakdown of unstable chemical bonds, releasing energy to power biological processes. Here, the pair of NADH and ATP contains the greatest amount of stored energy. Four groups of students carried out an experiment in which the effect of glucose concentration on the fermentation rate of yeast was measured. The fermentation rate was determined by the rate of temperature change of the fermenting mixture. Before beginning the experiment, each group practiced measuring the temperature of water and checked the group's thermometer against an electronic thermometer that gave a true measure of temperature. The following results were obtained during the practice. Accuracy is how close a measurement is to the true value, and precision is how close repeated measurements are to each other. An electronic thermometer gives a true value. Groups 1 and 2 are inaccurate because their values are lower than the true value. Group 3 is inaccurate because its values are higher than the true value, but it is highly precise, as the values are concentrated within a 0.5 degree range. Group 4 is both inaccurate, average of 19.3 degrees Celsius versus true value of 20.3 degrees Celsius, and imprecise, values spread from 18.0 degrees Celsius to 21.0 degrees Celsius. Each group conducted the experiment 3 times, Trial 1, Trial 2, Trial 3. 5 different concentrations of glucose were used in each trial. Each group plotted its results on a graph. The black bar represents Trial 1, the white bar represents Trial 2, and the striped bar represents Trial 3. In science, reliable results are those that are consistent, stable, and reproducible across multiple trials under similar conditions. The data from groups 1 and 2 are scattered more widely than those from groups 3 and 4, especially at the 10 and 15 marks. This means that groups 1 and 2 are less reliable than groups 3 and 4. If data are both precise and accurate, the results are inherently reliable. Therefore, stating that something is less reliable and more precise and accurate is a contradiction. Both Group 3 and 4 are reliable because their data are concentrated within a narrow range. However, Group 4 is more reliable than Group 3 because its data range is narrower. The endosymbiotic theory proposes that chloroplasts originated from free-living cyanobacteria that were engulfed by a primitive eukaryotic cell. This theory is supported by several key similarities. Genetic material, both chloroplasts and cyanobacteria possess their own circular DNA. Membranes, both have double membranes. Ribosomes, they share similar types of ribosomes. Function, both use chlorophyll for oxygenic photosynthesis. The light-independent reactions, or Calvin cycle, are the second stage of photosynthesis. This process occurs in the chloroplast stroma, where the enzyme RuBisCO fixes carbon dioxide. Using the ATP and NADPH generated during the light-dependent reactions, the cycle ultimately converts carbon dioxide into glucose and other sugars. In the light-dependent reactions, light energy is used to split water molecules, releasing oxygen and converting ADP and NADP into the high-energy molecules ATP and NADPH. The light-independent reactions then use this stored energy to convert carbon dioxide into carbohydrates. ELSA read that red algae survive at greater water depths than green algae because of a pigment in the red algae called phycoerythrin. This pigment enables the algae to absorb more of the green light available at greater water depths. ELSA decided to investigate this by carrying out an experiment. Using a standard technique, the single-celled algae were trapped in jelly balls. One set of balls contained green algae, and another set contained red algae. To measure the rate of photosynthesis, ELSA used a stopwatch and the pH indicator phenol red. Phenol red changes color in solutions with different concentrations of carbon dioxide. In low carbon dioxide concentrations, phenol red is pink, and in higher carbon dioxide concentrations, it is yellow. ELSA placed the jelly balls into test tubes and covered them with a solution containing dissolved carbon dioxide. Phenol red indicator was added to each solution. The diagram below shows the setup of ELSA's experiment. The experiment's dependent variable is the rate at which carbon dioxide is consumed, while the independent variable is the type of algae being tested, red versus green. Accordingly, ELSA hypothesized that red algae would photosynthesize at a faster rate than green algae under green light conditions, or in other words, they would have a higher rate of carbon dioxide uptake. Controlled variables, or constants, are the factors in a scientific experiment that are kept the same to ensure a fair test. By holding these variables constant, researchers can isolate and measure the effect of the independent variable on the dependent variable. In this experiment, the following must be controlled: number of balls in each tube, number of drops of phenol red, initial concentration of carbon dioxide, volume of the solution, initial pH of the solution, temperature, strength, and source of the light, and amount of algae. In this experiment, the independent variable is the type of algae, red versus green, as it is the factor being changed by the observer. The dependent variable is the time it takes for the color to change from yellow to pink, as this is the result being measured. ELSA's hypothesis would be disproven if the color change time for the green algae is either less than or equal to the time observed for the red algae. This result would directly contradict her prediction that red algae photosynthesize faster, and thus consume carbon dioxide more quickly. This is a very good suggestion. By setting up an identical experiment in the dark, you create a crucial control group. This control would help determine if the observed color change is indeed caused by photosynthesis, a process that is dependent on light, or if it is the result of another chemical reaction that occurs regardless of light exposure. Even if the additional experiment in the dark supports the hypothesis, there are still other plausible explanations for the results. There may be more living algal cells in the red algal balls than in the green ones, which would increase the rate of photosynthesis. Additionally, the phenol red itself might affect the rate of photosynthesis differently in the two types of algae. Finally, differences in the rate of cellular respiration between the two algae types could also influence the results. This section is worth 19 marks, which constitutes 15.8% of the total exam. It tests your knowledge on photosynthesis, experimental design, and cellular biology through a combination of multiple choice and short answer questions. Multiple choice questions, 8 marks, chloroplasts. Questions 8 and 14 focus on the structure and functions of chloroplasts. Photosynthesis, questions 9, 10, and 15 cover the inputs and outputs of the light-dependent and light-independent reactions. Scientific method, questions 11 and 12 assess your understanding of experimental reliability. Endosymbiotic theory, question 13 tests your knowledge of how endosymbiotic theory explains the origin of chloroplasts. Short answer questions, 11 marks, experimental design. Question 11 is a comprehensive question that requires you to design an experiment to test a hypothesis. This includes identifying the control, dependent, and independent variables. You'll also need to explain how a dark experiment serves as a crucial control and provide alternative explanations for the experimental results. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.